efficiently separate a mixture of DNA fragments like poly A, saDNA, DNA, 25, 50, and 100 poly 80 dsNAT, DNA, sorry, within 330 seconds, and a commercial DNA ladder comprising of 14 fragments within 800 seconds using vo- a sample volume of 1 microliter. The second, the second one is the pillar structures microfluidic chips. In this one, this investigation developed a PMAA PDMA hybrid microfluidic device that has an array of micropillars that etched in PMA layer. Here we can see overview of the recent research articles published since 2010, and we can see the type of chromatography, which stationary phase they use, and the microchip that this study is used, and the biomolecule that they try to separate and identify. identify. So, for example, this another group reported a silicon PDMS hybrid microfluidic chip for RNA extraction, and this microchip extracted dengue virus serotype 2 RNA from a pre-lysed virus culture within 30 minutes. This one is the resin beds incorporate in microfluidic chips. This group reported a glass PDMS hybrid microfluidic chip for selective ssDNA separation using AC principle. This microfluidic chip had three conjoined microchannel chambers, namely the isolation, transfer, and the enrichment chambers. So the first channel chamber is packed with human immunoglobulin E immobilized microbe for selective isolation of 87 mer ssDNA from a randomized ssDNA mixture. The second microchannel chamber contains solidified agarose gel that transport the thermally eluded ssDNA strands to the third microchannel chamber via electrophoresis. So the advantage of this microfluidic device over existing devices is the possibility to combine solid phase extraction and the electrophoresis on a single chip. Another one, Parton, another group, demonstrate sex identification on three ancient individual using an integrated glass microfluidic chip. The DNA samples obtained from their using their bone powder. Another group developed a disposable hybrid PDMS PCB inscribed with a copper electrode microchip containing agarose gel for DNA separation and detection. From 1 kilobase DNA ladder, el electropherogram. Another one is the monolith incorporate microfluidic chip. Another is the nanowires incorporate microfluidic chips. And this one that I found interesting is the separation of proteins using open channel microfluidic chips. Uh, Here, several different molecules have been explored to decrease non-specific adsorption of proteins on microfluidic chips during MC, MCE separation. This group coated an aldehyde functionalized hydrophilic polysaccharide dextran on a amine PDMS microchannel surface to decrease protein absorption. The glucose oxidase was chosen as a model protein to study the coating efficiency, and the dextran coating successfully prevented non-specific protein absorption even after five successive separation experiments. Another one coated a MMA microchannel with ethylene glycol to suppress the non-specific protein absorptions. Another application is using resin incorporated microfluidic chips. This group report a PDMS a microfluidic chip packed with mesoporous silica beds. The microfluidic chip efficiently separates dextran, a neutral polysaccharide, and BSA, relatively hydrophobic than dextran, based on their hydrophobicity with a retention factor of 3.2 and a resolution of 10.7. Uh, Another application is the monoliths incorporate microfluidic chips. Another group introduced a pre-synthesized mesoporous silica monolith disk that in the extraction chamber of microfluidic chips was followed by a monolith functionalization with octadecylated silane, a high protein like insulin, cytochrome C, lysosome, and myoglobulin, and hemoglobin. Absorption uh,
10 minutes.
of 94.8 to 99.7% was obtained using this microfluidic chip. Is well, finally, I can show you the conclusions. The future of microfluidic device for separation of biomolecules like nucleic acids and proteins from complex biological mixtures at analytical scale appears bright. The glass and polymer materials were highly preferred over silicon as the microfluidic chip material due to their numerous advantages. Researchers successfully used different technical approaches to pack chromatography resins within the microchannels. That's it. Thank you.
Good morning, everyone. I will talking about of this article called Parallel Channel Electrotaxis and Neuron Screening of C. elegans. They report a novel microfluidic method to conduct C. elegans electrotaxis movement assays and neuronal imaging on up to 16 worms in parallel. They use three strains. You can see here. They use three strains, one wild type strain and two fluorescence protein defective mutants for expressions in muscle cells and expressions in dopaminergic neurons, which causes neurodegenerative diseases. Here you can see the experimental setup used to perform this study. It consisted of four channel sections. First, branching channels for worm loading and distributions. 16 parallel electrotaxis screening channels. Tapering channels for worms immobilizations and imaging. And braking channels for unloading the worms. Um, Constant channel dimensions at each bifurcations were used to maintain the same pressures and voltage. Drop up to the electrotaxis screening channels and possible and ohms laws. Um, they use young adult worms were loaded into the microchannels using a syringe and pushed slowly until all the tapered entrance channels were occupied by worms. Next, the worms were manually pressure pulsed and placed into screening channels. To further demonstrate the applications of this device in chemical screening, the worms were exposed to C6OHD, that is a neurotoxin that has been reported to induce Parkinson's disease like symptoms by selectively degenerating the dopaminergic neurons. Um, Here you can see the time-lapse images showing electrotaxis of wild-type worms in parallel channels after exposure to this neurotoxin. The black arrows indicate worms responding towards the cathode, while MP arrows show worms with different phenotypes, such as uncoordinated movement and sudden freezing due to this neurotoxin exposure. The worms trade in the separate channels, and arrowheads are showing the dopaminergic neurons. They quantified worm speed, body event frequency, electrotaxis time index of responder worms, and electrotaxis turning time in the three strains. The electrotaxis swimming speed and body bend frequency were quantified using image software, and neurodegeneration was assessed by simultaneously immobilizing the worms using parallel tapered channels. This figure shows overexpressions in mutant strains of muscle cells induce significant decreases in the worm's average speed, but electrotaxis time index whereas no effect on the electrotaxis time turning time. And finally, the untreated wild-type strain and mutant strain for dopaminergic neurons exhibit normal swimming speed, and all the others parameter mentioned attribute to their health history, whereas the 6 OHDA or neurotoxin treated worms showed a slower response in terms of swimming speed, body event frequency, and electrotaxis time index. In conclusion, this work demonstrated an easily to operate, simple to fabricate, and reusable microfluidic device for the analysis of the electrotaxis response of multiple worms and solutions. Okay, that is all.
Good morning, colleagues. Today I would like to talk about this article called Multi-Cell Type Glioblastoma Tumor Spheroids for Evaluating Subpopulation Specific Drug Response. In this article, the authors create a system that models a spheroid of glioblastoma cells it's a multi cell line experiment. So what they created was two models. First, they produced a spheroid with two cell type. Here you can find the cell lines that they use. So first they use the wild type, which is this U87, and it's a glioblastoma cell. The second cell line is A172, which is also glioblastoma cell. And the third one that has a mutation in the epidermal growth factor receptor. So this is a characteristic of glioblastoma cells. The cells that have this mutation they are resistant to some of the treatments, as you will see further. And the astrocytes, which is a different cell line, but it is part of the environment that they want to replicate. So here you see that each of these cell lines is going to be labeled with a different fluorochrome. So you will see that the fluorescence of each cell line will display this fluorescence. All right, so let's see how they produce these organoids. They use a control with no treatment, and then they expose these spheroids to different treatments that you will see here. So the first treatment is dacomitinib, which is an inhibitor of the epidermal growth factor receptor, and this one is a second generation treatment. They also assessed the erlotinib, which is a first generation treatment, and it's also inhibitor of the epidermal growth factor receptor. And these are the two cell type spheroids, okay? So here you'll see that they have one glioblastoma cell line. In this case is U373, one astrocyte cell line, and the nuclei are going to be stained with DAPI. And in this figure, in the section B, you will see that they also have one glioblastoma cell line, one astrocyte cell line, and the nuclei are indicated with the DAPI fluorescence. So here we see that the control in the first row. In the second row, you will see the treatment, the result of the treatment after 7 days of growth, and then with the exposure to this dacomitinib in different concentrations. And in the third row, you see the erlotinib treatment, also with different concentrations. And as you can see, the erlotinib treatment affects to the to the glioblastoma cells more substantially than the dacomitive, as you can see here, because the astrocytes have proliferated, but the the glioblastoma cell line has been affected. And with the second spheroid formed by this A172 glioblastoma cell line, you see that they don't respond to the treatment. So in the next essay, they created the spheroids with 4 type cell lines of glioblastoma and 1 astrocyte cell line. So here you can see displayed in this pie graphic, in this, they show the distribution in the day number 7 and after the days of the treatment until the day 15. So you will see how the cell population has changed in this time period, and you can see how the percentage of each of the cell lines has diminished or increased according to the treatments. So in the first, in the A part, we have the control, B, the erlotinib treatment, C, the dacomitive, and D, the treatment with the P53 activator. And you can see how the subpopulations shifts. They also wanted to test the proliferation of the cells, and they used an ATP activity quantification assay, and also a live death staining and imaging over time in the culture. So you can see here how T1 is at the 7 days, at T2 represents the day 15. Okay, so here you can see how the percentage of total cellularity, how it varies in each of these cell lines. And also you can see these beautiful images from day 1, day 4, and day 7. And as you can see in the center, there is a hole, but it's of a limitation of the technique. It's just because the laser cannot penetrate to the center of the spheroid. And you can see that with the live death staining, we see that there are a few dead cells represented in red fluorescence, and the majority green cells are alive, as you can see here, how the spheroid has progressed. So these authors, they created this model which represents the complex dynamics of the glioblastoma cells. And it is going to, they are going to use, the further view is to use cells from the patient. Okay, thank you very much.
Hi everyone, I'm going to be presenting this paper by inspired nitric-oxide-driven nanomotor. Here, the authors report a nitric oxide-driven nanomotor in which medical use fluorescent hyperbranch polyamide and biocompatible L-arginine were chosen to synthesize hyperbranch polyamide L-arginine nanomotors. The carboxy group in L-arginine and the amino group in the upper hyperbranch polyamide are combined by electrostatic interaction to form these nanoparticles. The particle size of the nanomotors increases with the concentration of L-arginine, and the resulting nanomotors have a spherical morphology. Owing to the strict demands for biomedical applications, self-destroying nanomotors have been the ideal choice. So the particle size of these nanomotors in hydrogen peroxide solution decreases from about 170 to 70 nanometers for 18 hours, showing that it can self-destroy. They also did in vivo assays, and the presence of in vivo the presence of a metabolic enzyme nitric oxide synthesase can convert L-arginine to nitric oxide and L-citrulline. So in this work, they use hydrogen peroxide to simulate reactive oxygen species to investigate the nanomotor's movement behavior. Here you can see some time-lapse images of the nanomotors in 10 seconds. And these images illustrate that the smallest nanomotors cannot move in a hydrogen peroxide solution, while bigger nanomotors can move with the generation of bubbles on one side, with a straight movement light, even in different concentration of hydrogen peroxide. For nanomotors with a higher concentration of L-arginine and larger sizes, the bubbles are generated in several directions, so the movement line is a curve. They also study the uptake behavior of the nanomotors by MCF7 and HUVEC, and with confocal laser scanning microscopy, it can be observed that the nanomotors can move in the MCF7 cell environment with and without extra addition of hydrogen peroxide. And it can also be seen that the nanomotors still maintain a certain vibration after entering the cells due to the existence of reactive oxygen species, nitric oxide synthesized, and other active components inside the cells. So the results here indicate that the increase of concentration of reactive oxygen species and nitric oxide synthesized can promote the nanomotors movement, thereby promoting uptake of the nanomotors by the cells. The uptake behavior of the nanomotors by HUVEC cells under different conditions also display a similar trend, and confocal laser scanning microscopy and corresponding 3D reconstruction images were used to confirm the intracellular localization of these nanomotors. They also saw that cell viability of HUVEC after nanomotors intake gradually increases with the culture time. So to investigate the effect of the nanomotors on cell viability, the effects of different concentration of nanomotors on the growth of HUVEC and MCF7 were studied. Hyperbranch polamide alone has no effects, and L-arginine alone promotes the cell proliferation. So the addition of 50 to 100 microliters of nanomotors also promote the cell's growth with a significant increased cell density without cell death. However, excess of nanomotors can inhibit cell growth. These nanomotors can be used as a anti-cancer drug for its ability to produce nitric oxide continuously, especially because the amount of nitric oxide produced in MCF7 is significantly higher than in HUVECs. The amount of nitric oxide produced in cancer cells is higher because of a more efficient movement of the nanomotors due to a higher concentration of reactive oxygen species in these cells. The nitric oxide produced by these nanomotors after the enter cells can cause the calcium influx. And finally, in order to verify the universality of this formation mechanism for nanomotors proposed in the paper, 3 amino acid enriched organic compounds were used. 3 amino enriched organic compounds were used: chitosan, polylysine, and heparin with folic acid. And these were chosen to react with L-arginine, and the results shown here confirm this universality mechanism proposed, which can be extended to synthesize different kinds of nanomotors propelled by nitric oxide. So this is the paper. Thank you for listening.
Okay, this is my paper, droplet microchip with substance exchange capability for the developmental study of C. elegans. The worm has been used as a multicellular organism due to its short life cycle and its relevance in human genetics. Drop microfluidics is optimal for this study. In this article, a microdroplet chip is indicated to investigate the post-embryonic development of C. elegans in larval stage L1. these chips catch drones and can catch worms. In addition to investigating the development of C. elegans, the chip facilitates the study of worms in response to multiple drugs. In this part, the chip contains various functional elements, a control unit, a droplet trapping matrix, a worm collecting unit, changing with contain three liquid inlets, oil, worm, medium. The 116 drop trap, 16 rows with 10 traps in each node. And this part B indicates the procedures for handling the chip to generate a series of worms in the droplets. Fluorocarbon oil FC40 was used as continuous phase due to its biocompatibility and good solubility for oxygen. It shares the oil on the chip to maintain its hydrophobic properties. As the oil phase of the worm progress, it pushes the worms into the chip. And this part, 16 microvalves were used to control the flow between the network that connects to the drip trap. And the rows in the different rows could be formed without cross-contamination. The rest of the rows were controlled by adjusting the opening of the microvalves. The successive loading of samples indicates the operating stability of the microvalve. And this part, exchange of substance between a moving dye flow with rhodamine solution and a drop of deionized water. In part B indicates the change of rhodamine solution in a single drop during the exchange of substance. The exchange of substance was occurred in a single drop between 1 and this part indicates fluorescence in a row on 10 drops as a function of time. Multiple drop exchanges in a row were accomplished in less than 6 minutes. And figure 4, the L1 worms were separated into droplets on the same chip to maintain the cultivation of worms feed 3 times a day until they reach the adult stage. And conclusion, this chip is important to investigate development of worms from the L1 stage and allows you to manipulate the worms inside the chip in a controllable way. Thanks.
Can you see my screen?
Hi, Luis. Wait a minute. I have a message. I have. I don't know if I have good connection because I have to leave now. I'm going to pick to Aedo the incubator. So if I am lucky tomorrow, I will bring the incubator to Pacheco. Okay. I will continue with that now, but I don't know if it will be good inside the car. So see you tomorrow. Bye.
Okay.
Good morning. my paper, the Elegans colony formation as a condensation phenomenon. Uh, this, this article studies the behavior of C. elegans when they form colonies and compare the experimental results with a theoretical models inspired by physical modeling of molecules. This is experimental and computation. Uh, Theoretical model and simulation of computer is very interesting. Theoretical approach include fast separation, lattice gas models, and reaction diffusion models have been successfully applied to a wide variety of collective organismal phenomena, including formation, bacterial blocking, and biofilm formation. In this paper, it is used for study dynamical walls colony of Cl. In the figure 1 shows the formation of C. elegans colony with bacteria. They grow worms to adulthood and then wash it and transfer them to a fresh agarose plate in the absence of the added bacteria. For high density concentration of worm formed colonies, in contrast with low concentration worms did not form colonies. The figures E and F, E and F shows density of these dispersed downward worms. The density of the out-of colony worms increase linearly with the seeding density. The worms can be out of the colony or inside the colony, and one can ask the individual worms feels attraction by the colony. In this paper indicate not. The worms don't feel attracted for the colony. In these figures, the density of dispersal downward worms is linear and here and here. In downward worms and adult worms. In figure 2, A and B show the colony of C. elegans and the individual words. In the times, these words display movement to the colony are in a trajectory which is explode in these figures C. Figures C show on a time scale of minutes the trajectories of the out of colony words. Example, a random walk. Similar to the alcoholic man way to the street, on the street. Figure D, a logarithmic plot of mean square displacement versus time from 15 to 2000 seconds has a slope in this case and this case. And a slope of near to expected value for the random walk. In the random walk, this slope is near to 1. In contrast, this is the random walk. And in this case, the plot corresponds to the displacement of the board and in the initial motion in a few seconds in decay, which correspond to that picture here 0, 2, 4, 6 seconds. Okay, and in this case the words show that the motion is near to ballistic trajectory. The slope is near to value 2, in contrast to the world inside the colony and displacement with the colony and the random work. One can conclude that the world's displacement more relaxed when they are in colony. But the C. elegans trajectory are not biased toward colonies. When colony formation could be due to Caenorhabditis, with the words moving preferentially toward a nascent colony in response to the gradient of an attractor. In these figures, the word is the blue curves, and the colony corresponds to the disk, orange disk. And they calculate the velocity, the radial velocity and tangential velocity, and they plot the speed or velocity versus distance from center of color. In these figures, we can see that the words are not attracted by colony because the radial component of velocity not is more greater than a tangential velocity. In few words, radial velocity is more or less equal to the tangential velocity, and it can show that the colony don't attract the water. In these figures, the symmetric polar plot of tangential component versus this distance from center, it's evident that the taxis model for colony failed. In conclusion, the colony don't attract the world, and the world move toward the colony in random walk. The theoretical model is according to the experimental result, and is good model to describe this dynamic. Thank you very much.
Thank you, Luis. You have a questions?
I have a question for Giovanni. I really enjoyed the way